Hello, thank you to see my presentation today. And of course, thank you very much, Andy and Tom, inviting me to present important findings at your most important conference. So, my task is to tell you key issues, what happened in the history of science and virology, which is, of course, very strong connected to the story of humankind, as you are going to see. When analyzing what virologists are doing in reality, and we are going to answer the question why they are doing what they are doing. And in analyzing this, we are learning about the conditions under which science is working and when this has been established already 2,500 years ago. And when we understand that we have an inbuilt framework of misconception which doesn't allows us to see reality of biology in medicine as it is, then it becomes clear why they are acting, how they are acting, and how to escape out of this situation, which is way dangerous for everybody who is believing into this concept. And to make it short, it's easy like this. If you are believing that molecules inside your own body are turning themselves into a terrorist group called cancer, and that this terrorist group is going to eat you up, fighting against you, and very often is winning this war, then you also believe in the concept that this terror group is able to move inside your body. This idea is called metastasis. And if you are believing in metastasis, you are believing, of course, in flying metastasis. And that's the challenge we all have and which we are going to solve with this conference and with our activities to get a deeper understanding what's really going on, and of course to get rid of the fear which our thinking is forcing us into. And myself, I came into this field when I was a biologist doing research in marine biology, and I came across marine algae which was not producing spores, which you can easily see, but instead of producing spores, producing much smaller structures, mini spores. At this time, when I saw the pictures in the electron microscope, I saw thousands, millions of those particles, and they look like we are thinking viruses do look like. And so I believed that I have discovered, isolated, a harmless virus. Because the algae producing these particles, here in this picture you'll see an enlargement on the right side. On this next photograph you'll see the isolated particles. I thought I'm the first to have discovered and established a stable virus-host relationship. So I became virologist. And even so, having something in my hand that was not doing harm, but is in the end a useful structure. On the other hand, I believed in dangerous viruses and were teaching students to take care in front of HIV and better using double condoms than nothing, no ones. In the end, I learned to characterize such a structure, isolating it, making photographs inside the organism, and isolate the particles outside the organism, and did biochemical analysis of its components, the proteins and the nucleic acid. And quickly, I was informed by an Austrian professor that something is wrong with HIV and AIDS, because he asked me if I did check the evidence, and I told him, no, the whole world is saying that HIV does exist and it's dangerous. And he said he was not asking the world. And I asked him, why you are adopting this idea? And he said he heard some rumors that something definitely it's wrong in this field. So he asked me to go into the library and to check the evidence on HIV, which at this time had another name, on my own. And so I came into this field and I quickly realized, step by step, that in order to isolate a particle and to have it in the hand, that they went into electron microscopy and they are presenting us photographs from dots of tissue where everybody sees the big part of the dot of the tissue and smaller particles, claiming those particles do represent viruses. But no control experiments. If those particles would be spherical and not belongings of the tissue dots, like documented here, then it should be seen that when you do a series of electron microscope around the sphere, that you'll see nothing, and then you see a smaller circle, a bigger circle, then a smaller circle again, and then nothing. Whereas in all photographs you always see a part of a tissue and only particles which are spherical. And this, it was very clear to me, where I isolated a structure here and showed that they are present in the organism, in the marine algae. And I really quickly found that none of the electron microscopic photos representing viruses are inside organisms. There is no single photograph on Earth saying that this particle has been found in a human being, in an animal, or in a liquid of this person. And of course, there is no presentation that they are isolated in the meaning of the word isolation, and no biochemical characterization of its molecules. And as I showed you, I spent a lot of time on the electron microscope, so I know all those techniques, and I quickly realized that they are showing us things which were not isolated, but were a typical part of tissues dying in the test tube. And from those photos, they make a model, for example here the measles virus, and claiming this represents a virus. Whereas they never isolated something and they never showed the whole piece. And to get even worse, what they are doing, since the genetic area, since it is possible to multiply nucleic acid, to sequence nucleic acid, what they are doing, they take small parts of nucleic acid out of dying tissues, they sequence them, they determine the composition, and just mentally using software programs, they make a strand out of single small pieces, and the product at the end, they are naming a viral genome. And this is easy like this. And it's easy to show that it's wrong and that's a misconception. And this is a self-fooling, and the virologists are fooling themselves and they are fooling the public. It's not on bad intention, it's just because they are completely anti-scientific. They never ever tried to question themselves, and they never ever followed the first rule in science to perform and to document control experiments in order to show and to exclude the possibility that the results are the product of the techniques what you are using. So I'm able to state here right and now that all virologists claiming viruses which should cause disease are anti-scientific. Therefore, the laws applying corona means or the duty to get vaccinated are not justified because they all are done under presumption that there is a scientific work out there. Therefore, all the laws on infectious diseases are violated by the fact that there is no science, but there is pure, easy to see, easy visible anti-scientific behavior and self-fooling. So how come in the history that this is able to happen? Where's the origin of this misconception? And as I said in the beginning, we have to go all back 2,500 years to the Greek philosopher Democritus. And he did what the steps and was successful in introducing the idea that life from this time on is only to be explained by pure materialistic interactions of molecules, and they excluded systematically the action of consciousness, of a field, of a spirit, of God. And it was systematically excluded from science. Why? They had a good justification. He said, and his teacher and his students, Epicure, they said that the religions always posing fear upon the people in front of God, claiming that he's punishing the people with disease for their sins, and we don't like this, and therefore we are establishing a view on life which only material interactions, we do only consider material interactions. And this is the materialism which we have since 2,500 years. And this kind of thinking is forcing us directly into believing that there are materialistic carrier of disease, a toxin of disease, Latin, virus. And one very favorite who criticized all of this is Plato. Plato is citing his teacher Socrates, and Socrates is citing somebody else who he heard about, that he was criticizing and saying that since this time the Greek medical doctors are not able to deal with diseases. When something happens with the eye, they are treating the eye. When something happens to the bone, they will treat the bone. But they all forgot that life all comes out of a soul. And so when a person is diseased, then the soul is to be treated. And Plato says something very cool. He says that there are two medicine bodies they do exist. The one we know, and he says there we use medications to suppress symptoms, and we use it for the unfreed, the slaves, so they getting back soon to work again because we suppress symptoms. And there is the medicine for the free, and this is where the soul is treated. Very interesting what Socrates is saying, and I just use this opportunity to mention this very important book of O'Mahony, Can Medicine Be Cured? A Corruption of a Profession. Here he claims that the pharmaceutical companies are responsible, that the whole field of medicine is completely corrupted and it can't be cured. It's going to kill everybody, and only a war or a humanitarian catastrophe can cause a reset. Now I'm happy for Corona, because Corona is the reset of this medical system. And funny enough, he believes in all these concepts of antibiosis and of vaccination. But at the end, on page 262, he is stating that there are two medical systems. The one of Aesculap, where symptoms are suppressed with medications, and the another one, where do I have her? Hygieia, where health is defined as the harmony inside yourself and with your environment. And that's very, very strong. And look, the snail ear represents our nervous system, you know, and it's not here where the word is important, because a word can kill, a word can heal, a word can hit your bone, a word can hit your stomach. Here, she is feeded and not her poison taken out. Very interesting about Hygieia. O'Mahony is speaking in his book and said, health is the harmony inside yourself and with your environment, but that this medical system never had a chance. But I can tell you, this medical system existed till 2,500 years ago, and this was discovered by a German medical doctor, rediscovered in 1982, Dr. Rieke Gerd Hamer. If I have time in this presentation, I will inform you about his most important findings. If not, probably I do present this in a workshop, or you'll find it in my information available on my web page in English. Let's come to another German medical doctor. He was the one who sharpened the existing idea that life is only explained by mechanical interactions of molecules. And he is the one who sharpened it in a way that he just claimed out of the nothing that life do exist only in single cells, and that every organism is coming into existence by cell division. And this was not true at this time. This was violating every finding. I will quickly show it to you. But he said it because he had to become rich. It's easy like this. His father had 200 million euro debts and didn't allow him to become priest, because he said, look, you have to deal with my debts. And so he forced him to study medicine. And there's a long story which is of greatest importance on him, but now only the essential thing. Whereas in the year 1848, at the German Revolution, which failed, completely failed, we had a backlash, a rollback, he tried to fuse medicine and state with very humanistic statements. He said, look, all those plagues do only come into existence because the people have no toilet. They live onto their feces. They have no chimney. They have no dry heating material. They have no washing possibilities. They have insects in their beds, in their dressings. Therefore, they become all the skin rash. If they live in normal conditions, they get healthy quickly. And if they're in healthy conditions, they never won't get ill. And these true statements from 1448, for this he became famous. But he failed with his attempt to unify state and medicine. But he succeeded 10 years later in 1858. And there he came up with a completely different model. And this model he was naming the cellular pathology. First claim that all cells, all organisms come into existing through cell divisions. And second, that every disease comes out of a single cell in order the cell is producing the toxin of disease, Latin virus. So he re-established this idea that the toxin of disease, which is built inside the body itself, it's going to spread and to multiply. And it's really dramatical, because already from the year 1650 on, it has been proven that the theory of a virus, of a toxin of disease, which is spreading from one cell to all the other cells, this is completely falsified by the observation with lenses, later with microscopes, that when a tissue, and all our organs consist out of 4 different kinds of tissues, when an organ becomes ill, then the change is only in one tissue, and it's not spreading right and not spreading to the left. And this was suppressed by Virchow in order to come up with his cellular pathology, which, by the way, was rejected by the American science for 1 generation. After this, after 30 years, the youngsters took up this new revolutionary idea, and we are all in the same trap, in a mind setting which is completely wrong and which does not allow us to understand what we see, because we interpret it systematically in a wrong way. Also, the claim that life comes out of one cell and there are only materialistic interaction is forcing us into belief that there is a genetic material directing all the molecules in order to build an elephant, a tree, or you, the ones watching this video. We are forced into the thinking that there is a genetic material. It's a logic which is there, but which is not the truth. And imagine what was published in a German newspaper, Die Zeit, in 2008, with the title Genome in Dissolution. And here you will find the completely rejection of the notion that we have a genetic information, and of course, most important, that we have viruses, because viruses since the year 1954 are defined as mini chromosomes, as smaller parts of genetic material which are very dangerous and which are enveloped or not. The pure one, Ebola, are naked, of course. So this is what we are forced into thinking. But the reality is what you will find here in a German newspaper published in the year 2008. Here is a translation. The genome was considered to be the unchangeable blueprint of the human being, which is determined at the beginning of our life. Science must bid farewell to this idea. In reality, our genetic makeup is in a state of constant change. 2 years ago, 25 geneticists sat down at the University of Berkeley, California, at Berkeley, to answer the seemingly simple question, what is a gene? However, the attempt to define the basic concept of their field precisely proved to be extremely difficult. The expert meeting almost ended in disaster, recalls Karen Elbeck, professor of human genetics at Berkeley and host of the Round Table. We had meetings for hours. Everybody was screaming at everybody else. And let's see the argument. The argument at Berkeley has little to do with research readiness. It was the first symptom that the life sciences, as yet unnoticed by the public, and recall, this has been published in 2008, and it's still not known to the public that the whole field of molecular genetics is disproven, and with this the whole field of virology. Because why? Because our whole economy, our whole money system is depending on the idea. Since 50 years, we invest billions of dollars into the idea that we are changing life with molecular genetics. We have molecular genetic tests, we do PCR, but this is all fundamentally flawed and disproven. But they can't give up because it became a big business. And this is one of the reasons of Corona. So periodically, when a new plague was able to construct in the media, then all those investors take off their money, and they have to. If not, the whole economy system would implode. So Corona, that's a good, another good aspect of Corona, is stabilizing our money business system, our financial system. So let's go back into the text. What researchers are bringing to light in the chromosomal strands of humans or animals goes beyond the previous thought patterns of genetics. Similar to the beginning of the 20th century, when Einstein and his comrades in arms formed a new physical worldview, the age of relativistic genetics may be dawning. Medical research in particular is facing new challenges. The first outlines show that body and soul, their health, disease, development and aging are subject to a genetic interplay whose complexity exceeds all previous conceptions. So, and here we have already one example. Science failed to tell this fact to the public. So they continue. Now they say, look, we need another 40 years, another 40 billions of dollars and whatsoever. And now we study the interaction of environment, proteins, unknown factors with the genetic material. And this idea is called epigenetics. It's ridiculous. So here you will see very clear that we have a severe dishonesty of scientists who are aware of these contradictions but not telling the public. So still people out there believing that we have genes, we have gene technology, we have an mRNA vaccination, but it's completely a disproven model. Let's read. Continue. Medical research in particular is facing new challenges. The first outline shows that body and soul, this we had already. So, but now the essential sentence. Geneticists must abandon their image of a stable genome in which changes are pathological exceptions. So, what they did, they constructed a genome, of course with mathematical means, using billions of small nucleic acid molecules, determine their sequence, and then put all the sequence into programs and form a big sequence, which they call the chromosome. But this does not exist. This is how they are forced that there must be a book of life, there must be a genetic material, when always out of a given seed, always a tree it's coming from. So there must be a genetic material. We are in a trap since 2,500 years, forcing us to think in such a materialistic explanation. So once they determined the genome, of course in a computer, they never found it. Every exception they found afterwards, of course in disease they make a biopsy when something happening with the breast or wherever, and they analyze the nucleic material and they see, oh, it looks different. And this is what they're calling a mutation. And this you'll find in this sentence. The genome of each individual is in a state of constant transformation. So all what they are finding and claiming as mutations or as a virus, it's the normal product of the ever-changing of nucleic acids. So, as a result, every organism, every human being, even every cell of the body is a genetic universe itself. Here you will find the explanation. And with these findings, the whole field of virology is disproven as well, because they claim a stable genome of a virus, and in reality doing the same what the geneticists are doing. So we can say that the virologists are victim of the geneticists. But we are all victim because we are all in the same movie, or we are in the same cave Plato was describing, only seeing shadows on the wall and not wanting to turn around where the light is coming from, and even not trying to get out of the cave. So this is Plato's explanation, and we are all in. And look at this most important German thinker I'm aware of, Eugen Rosenstock-Huessy. He lived in United States because he had to escape of Nazi Germany. And he's such a deep thinker. It's incredible. I can really recommend you to start to read his books. It's really, really far away and mind-blowing, and it gives really a good orientation. And this man already in 1929, this was published in a book in 56. Then he died in 73. And in 2008, this book finally appeared in German in the way he wanted it to get published. It's not translated in English, unfortunately, but you will find a very good conclusion and description about the most important findings in this book, Speech and Reality, published at Ergo Books, 1970. So, what he wrote down in 1929. The place where there is room for growth in the scientific age lies between a new pair of antagonists, namely between research and knowledge. So, there's an antagonist, knowledge, research. The struggle between these two opponents remain largely unexposed. Scholars are quite clever and completely incapable of embracing the overthrow of their intellectual virtue. They are officials of the scientific order and always stand in opposition to the amateur. Science, of course, officially research belongs to official science, just like the Holy Spirit belongs to the Church. There is a massive amount of pseudo-research competing with the progress of free research. And the former is the only research that is faithfully supported by the official agencies and foundations, because it's the only research that seems worthy of support by the professional science officials. And think about this sentence written down 29. It researches cancer according to the outdated ideas of Louis Pasteur as if it were rabies. This is a double critique on the infectious disease, which brought automatically, as a result, the thinking on cancer to birth. Another giant in thinking and arguing is the Croatian philosopher Ivan Illich. You definitely have to read his book, Limits to Medicine. He published this book in 76, and he discovered that medicine, being part of the economy, are forced to produce every year a certain surplus, a 5%. And they can't find this in the human nature, that every year 5% people get more ill. So what they automatically are doing, they are widening their diagnosis and they are widening their treatment options. And in the end, they are doing harm to everybody. The human beings, the animals, the fishes, the water, and the soil. And this predicted in 76. He predicted Corona, that this is going to lead into a collapse, which we have. But thanks God, we have the means in our hand that this balloon, which definitely would go into a collapse of Corona, we do let it implode, and this you can take for granted. And we'll see soon that we, with our activities, do let this balloon implode. Why now? Exactly. Virologists are believing that viruses do exist and do look like bacteriophages, which do really exist. And bacteriophages are seen as the viruses of bacteria, but they aren't. A bacteriophage can't possibly do harm to a naturally found bacteria. Only if you keep bacteria very long in the lab so that they lost all their abilities, and all of a sudden, if you treat it bad, and they have no time to build really spores, which we know, for example, in chalk, then they build the mini spores. And such a thing I found more than 30 years in the sea. And I thought this is a harmless virus. There are mini spores which do have the ability and the task to have other organisms in order to better grow, because they do deliver nucleic acid, which is primary function, setting free energy, and they do deliver proteins. And they do have the capability to grow quicker, to grow on their own. And therefore, virologists in Marseille are fighting that those giant viruses, which they are called now, which they find in huge numbers all over the seas, a huge amount of biomass, they are fighting that this is going to be considered as the fourth kingdom of life beside the eubiotics, the real bacteria and the archaebacteria, the real bacteria and the eukaryotic cells, that those structures are considered to be part of the fourth kingdom of life, because they find they have metabolism and they grow and they get larger and larger and form new organisms. And such a structure I found 30 years ago in the sea. And then coming to see the contradictions in HIV, then I looked to the all in the field of virology, and I found this pattern that they are orientating themselves at bacteriophages, which are produced when bacteria are all of a sudden are not able to live anymore and have no time to build spores. And then they build their mini spores. And it's not an act of destruction, because all biomass of the bacteria, all its genetic material is retransformed in a highly coordinated process of metamorphosis into equally built nucleic acid, always the same size, always the same sequence. And they are real. And virologists orientated themselves at those bacteriophages we do really exist. Here a small detail, what you definitely have to know. In 52, virology, the old virology, so we had an old virology, and since 54, a new virology. The old virology gave up themselves doing control experiments, seeing, also from very healthy tissue, if we let this into decay, at the end we'll find the same molecules, the same proteins, which we consider to be viral. So they published this and they gave up themselves. And at this time in old virology, before 52, a virus was considered to be a protein with the capability to self-reproduce, because till this time it was believed that the genetic material is a protein itself. And only doing research with those bacteriophages, it became clear a protein can come only into existence if there is a catalytic acting molecule of nucleic acid. It can't reproduce on its own. It always needs, as a catalyst, a nucleic acid. And from this nucleic acid on, all of a sudden they believed, this must be the genetic material. So from this time on, they built a model, and finally Billy Boy, I don't mean Gates, I mean Clinton, he said in 2000, we published, we were reading the books of life. But this was completely, at this time already, it became known, and it was clear, as you see in 2006, that it's completely mistaken, completely, and that the chromosomes published are an artifact built in silicio, in the computer, with software built out of ever-changing millions of smaller molecules. And when this research on bacteriophages showed that nucleic acid is needed to produce a protein, then a virus all of a sudden was, okay, a virus then is a piece of genetic material which is replicating always itself, and they orientated themselves how bacteriophages, which do exist, look like. And from this moment, they were deeper in the trap. And the one who was driving them with high speed to the wall was a bacteriologist, John Franklin Anders. He took over the idea from bacteriology. Oh, when we could destroy bacteria in the test tube, and when they transform themselves into phages, the viruses of bacteria, then probably we can do it in a similar way when we use tissue from animals or human beings, adding seemingly infected material, blood or salvia, spit, from people with measles, And then when they are dying, this tissue, this could be a sign that the material is transforming itself into viral matter. Of course, they never ever were able to isolate something, a structure, complete structure, even not a complete nucleic acid, but they just believing when in their test tube tissue is dying, this tissue became viral. And so they take single molecules out and they construct their models of viruses. But it's just a mental construction. They never isolated and they never were able to show structures they find in dying tissues, that they find the same structures in a human being or in spit. We all have to wear a mask, or we should, I'm not. But look, they never found a virus. They never could photograph a virus in spit. They never could find nucleic acid, which is the one which they presented us as viral. And this is all based on this single publication of John Franklin Anders, published on 1st June 1954. And he describes that he is treating the cells with highly toxic antibiotics in order to prevent that bacteria, which are everywhere, are killing the tissue. So if the tissue are dying, it must be a virus because bacteria can't be. It didn't come to his mind that this antibiotics he were using is killing the cells. And it didn't come to his mind that the reduction of 90% of the nutrition, so that the cultures are hungry to swallow everything and to swallow the virus and get infected, that the hunger is the cause of the dying tissue. So he never performed control experiments, but he was warning constantly in his paper. In doing so, however, it must be borne in mind that cytopathic effects with so superficially resemble these resulting from infection by measles agents may possibly be induced by other viral agents present in the monkey kidney tissue or unknown factors. He is repeating his warning. Several times a second agent was found. Obviously, the possibility of encountering such agent in studies with measles should be constantly kept in mind. Accordingly, the results that are summarized here must be subjected to most critical analysis. Or he concludes, two experiments essential in the establishment of the relationship remain to be carried out, because the evidence is only indirect. These will consist in the production of measles in the monkey and in the man with tissue culture materials. And these experiments never ever have been performed. Why? Because Anders became the Nobel Prize, was rewarded the Nobel Prize half a year later, but for a completely different thing, for an idea he had in old virology in 1948. But getting the Nobel Prize, this speculation, we himself named a speculation, this became a scientific fact and became the unique basis of the whole field of virology. And believe it or not, in the measles virus process in Germany, which I finally won at high court level, it is written in the sentence that this paper doesn't contain a scientific proof for a virus, and it is stated that is completely unscientific because no control experiments in this paper and all the other following papers are there. So therefore, this is completely anti-scientific, what they are doing, and there is no way to suppress the people by laws, by order, because the way basis science, it's not given, it's violated. And we have since 1998 the Safeguarding Good Scientific Practice written down. And here, every researcher who is receiving tax money has to follow these laws, these rules of science, and control experiments to exclude artifacts, to exclude that you are self-fooling yourself and fooling the public, are central part of the scientific method in all fields. And they are never performing this. And control experiments are easy like this. I performed them in the measles virus process. Now again with corona, this part already is published, that we are stopping the nutrition, we are putting the antibiotics, and the virologists are adding seemingly infected material, so the tissue is dying and they claim a virus is present. And with the molecules, they construct models and the genetic material. And we did the control experiments, we stopped the nutrition, we added antibiotics, we did no infection whatsoever. We did exactly the same what the virology is doing, and they are dying exactly in the same manner. The same thing we did with the nucleic acid. We let normal tissue die in the test tube, exactly the same way virologists are doing. We take out the nucleic acids and we add them up on each other like the virologists, and we came up with exactly the same results virologists are presenting. And believe it or not, this is the paper, the single paper where the genome of SARS-CoV-2 has been invented. And they describe step by step how they did invent the virus in a computer program. And if you follow this, you will see, in the nucleic acid out of a human being, they find no genome. So they do multiplicate it in high number with PCR, they don't find a genome. So again, they do a second step of PCR multiplication, adding 20% of known sequences into this mixture, and then they came up with a computer program with the genome of this virus. And you don't find the word control experiments in all of these papers. All other virologists, they just repeated the steps what they were doing, and they came, of course, to the same conclusion, the same result. So the public and probably themselves are believing, yeah, we just found the viral genome. They didn't find it. In humans, in the liquid, they had to do 2 steps of extreme replication of small molecules and then to apply a lot of software programs, and in the end coming up with a viral genome. And I skip now the details inside the publication of them. You'll see the contradiction themselves, which they are not aware, contradicting their own claims. To end my presentation, I have to go back to Virchow with his cellular pathology, with the findings which were known already by this time. And this will lead me into this field of body and soul medicine, which that did exist 2000 years ago and which was rediscovered by the German medical doctor Ulrike Gerdheimer. I'm following this research with my colleagues, and we do basic research in this field. He found that when you have an alteration in some of the tissue, be it the skin inside somewhere else, you always get a specific signal in the brain, and with this you can find out what happened to you at what time and what causes it have on your body and on your mind. And I found symbols for this, which simplify what it's going on in the 4 different kind of tissue we have. This is the hero. This is the man who for the first time in 2,500 years of our history did exclude the thinking of good and bad from biology and medicine, which we are forced into the belief that there is a good and a bad, because when we can't explain misbehavior, aggression, and also alteration and suffering and death, if we don't understand it, we are forced into thinking of a bad. And he's the first one who took out this notion with his finding, which in fact is a rediscovery of the things Socrates described. And he is describing on page 262 when he states that this medicine, which Rike Hammer rediscovered, had no chance. It had a chance. It was applied. You'll find it in every cultures. And this is what we are doing in order to make this public. I'm working on a scanner of the brain where you'll get easily those signals out, because Hammer found when in certain areas of the brain you have activities due to traumata lasting on you, that your behavior is altered in a very specific way. So if you don't understand it and your environment doesn't understand it, they will judge on you. And if you understand yourself, you are not forced to judge on anybody else. And I think this is of greatest importance, that humankind finally has a jump in development out of this good-bad notion, out of the notion that the war is the father of all new things. No, it's life itself, it's the principles we'll find, it's always symbiosis, it's always to try to go ahead and to forgive. A colleague of mine wrote in his book, The Sources of der Göttlichen, of God, that these findings, what Hammer found on individuals, this happens equally in our history, in societies, and this is actually happening at the moment with Corona. I'm very optimistic that we are going to overcome Corona soon and use it as a change in humankind for a better understanding of ourselves, of life. And therefore I'm working, therefore I'm grateful that you offered me this space in your conference, that you are watching this, that you are reading about this, that you're acting accordingly. We're acting in our way, and you will see, I'm sure, this winter, Corona, it's gone. You will see, and I'm sure. Thank you very much. Bye-bye.